So Mendel, you've heard his name before. You know about him. He was a monk. He was an Austrian monk. And we kind of consider him the father of modern genetics because of what he did with pea plants. What actually happened was he turned out to be good enough at monking that he got promoted to like head monk of his monkery monastery. And he inherited the kind of like gardening duties, I guess, from the previous head of the monastery. And this person had been real intense about his pea plants. Like the person before Mendel had organized pea plants in like very OCD ways. Like there was a garden of just all purple flowered pea plants, and then there was a garden of just all white flowered pea plants for like no reason he could figure out. And he found these traits almost kind of already organized for him in pairs. And so, of course, the first thing he decided to do was mess with them, start to actually cross them. Here's where we have to take a brief detour into plant biology because not all of you know this, and everybody should. Are you aware that plants have sex? Okay, some nods. Good, good. Plants reproduce sexually. That is how they reproduce. Now, some can also do asexual, like if you plant clippings from a plant, you're basically asexually reproducing that plant. But primarily, plants use sexual reproduction. Now, obviously, it doesn't look a whole lot like what you're used to, right? Because basically, because a boy plant can't like walk up to a girl plant and ask her out. Like they're stuck. They have to do this very much long distance. How do they get that done? Through pollen. What's pollen? This is going to get weird for some of you.
Is it just sperm?
Pollen is plant sperm. Yeah, pollen. Every pollen grain has 2 little plant sperm inside it. And what happens is, one way or another, the one plant has to get his pollen grain to the other plant's female part of their stuff, and then the pollen grain grows a tube down to the egg, and the 2 little sperms swim down the tube and fertilize the egg. What are the organs? We don't think of them as organs, but what are the sex organs of plants? Most plants, not all. Most plants. Most plants on Earth. Gametes are the cells they use, just like we do. Sperm and eggs, they've got them too. Where do they have those cells in most plants? How about flowers? Flowers are the sex organs in plants. Really kind of makes you rethink certain things. Giving other people flowers gets kind of weird when you think that way as a biologist, but don't think too much about it. Also makes allergies weird. The fact that you're allergic to plant sperm. Welcome.
I
know, but that's what that is. Okay, so a lot of plants get this done via animal intermediates like bees, right? Transferring pollen. Some of them use wind. What a lot of them have that is weirder is that a lot of them are able to actually reproduce sexually with themselves. And it does count as sexual reproduction because they're still shuffling up the genes and making sperm and eggs. It's just that the plants that have complete flowers, the sperm can fertilize the egg from the same plant. And so that's what Mendel is doing. I want you to picture him kind of stalking around his little pea gardens with a paintbrush and a pair of scissors, cutting off the male parts of flowers so that he controls the reproduction, and using the paintbrushes to dust the pollen onto the flowers that he wants to be cross-pollinated or self-pollinated. He's totally controlling which ones mate with which ones. And basically, what he does is take these true breeding gardens that he has, that his predecessor has kindly organized for him. True breeding just means when you self-pollinate them, you're always going to see purple flowers or whatever trait you're true breeding for. So he takes those, and he's got stocks of purple and white flower color, short and tall plant height, and yellow and green pod color, and he crosses the 2 versions of every trait. He cross-pollinates them. He's expecting, of course, something in between. Lavender, medium, greenish yellow. What does he get instead? He discovers that in each case, one of those traits very much wins, right? One of those traits shows up and the other one appears to be gone. It's a famous enough set of experiments that each generation has a different name that every geneticist knows. You say P generation, geneticist thinks, okay, true breeding, we'll find out homozygous later. True breeding plants that have, that self and come out with the same stuff. The F1 generation is what he gets when he crosses each stock and he gets all one color or trait. And these are called the F1. That's Latin. The F is for filio, which just means offspring. Elijah, you had a question?
No, sure.
Huh?
Sure.
You were just scratching your head. Awesome. Then he takes the F1s, the ones that all look like one parent and not the other, and he self-pollinates them. First set of conclusions. We didn't do that yet, right? Okay, first set of conclusions he draws from these experiments is that, first of all, there is something called a gene. He's the one who coins the term. That there is something physical in our bodies that is being passed to our offspring that is controlling the expression of these traits. You're getting something from your parents, a physical thing. I don't know what it is. I'm going to call it a gene. The root there, by the way, you might recognize. Gene means to create, as in Genesis, generator. Geneva? I don't know. Maybe. Second, that there are different versions of traits. That a trait can come in different related versions, like purple and white flower color, and that the different versions of those genes, we're going to call them alleles. If you don't know that term, that's one to get to know real well. Alleles are the different versions of the same gene. Some alleles turn out to be able to cover up other alleles, which becomes important when you realize, as he did, that you must have how many alleles for every single trait in your body? How many alleles do you get? Cole? 2. And how would you know that? Why would it be 2?
If it shows up like in a generation past, there isn't one like how Mendel knew it.
Yeah, how did he draw that conclusion? Why would you have 2?
Because in the F2 generation, if it shows up, it can't be one allele. If it was one allele, it would just be the exact same person.
Okay, 1 out of 3.
Or 2 sounds better.
2 is right. There's a really good reason for it, Danny. Because 1 came from each parent. Yep. Cool. You weren't wrong. It's just go back to basics, and yeah, you got 1 from each parent. So those are his first, most basic conclusions. Then he takes the F1 plants, self-pollinates them. That's when things get interesting. He's starting to understand this, but he's still really pretty much expecting, all right, I understand dominant now. I take these purple plants, self-pollinate them, just going to see purple. Not what happened, right? Surprised himself again. Real quick, to just look at what's going on under the hood, you remember how we depict alleles, right? What do we use as symbols for, say, dominant and recessive alleles for a trait? Yep. So what we were working with at first here, these true breeding stocks, were two dominant alleles being crossed with two recessive alleles, right? The true breeding means they're the same. That no matter how many times you cross this thing with itself, it's always going to be dominant. This one's always going to be recessive. So he's crossing these, and what he's getting is all big letter, little letter, right? All dominant and recessive together. You know the term for that? Good. Heterozygous, homozygous dominant, homozygous recessive. We'll write those down in a minute. Now he crosses the F1s with themselves. In other words, purple heterozygous with purple heterozygous. He's expecting all dominant. What does he get? He gets more purple than white.
Some white, some purple.
White comes back. White comes back at about a quarter of the offspring. Nice thing with plants, doing genetics with plants, is that you can make a lot of offspring, right? Every single pea in that pod is a potential new plant. It's a little pea plant embryo. So he's planting a lot of pea plants. And the more he plants, the closer he gets to this 3-to-1 ratio. Because that's what that is. That is a 3-to-1 ratio. 75% to 25% is 3-to-1. And he called these the F2 generation. So P is the true breeding parent. F1 is the heterozygous first offspring. F2 is the 3-to-1 ratio of dominant to recessive. Everybody okay? Is still following? Wherever I have pictures, that's just to help you visualize it. You feel free to use words instead of pictures as you take notes. Second set of conclusions come now. What he basically has learned is that those pairs of alleles, they have to be coming apart. Those parents have to be basically taking the big P and the little p that they had and going and pulling them apart and either giving one or the other to their kids, right? There has to be what he calls a segregation of alleles. He calls it his law of segregation. Under the hood, we know that these guys were heterozygous. About half of these were heterozygous. Half the offspring end up being heterozygous, a quarter homozygous dominant, and then 1/4 of them homozygous recessive. His second set of conclusions. Number one, those alleles in the parents have to be being separated. He even correctly guesses that the time that segregation happens must be when sperm and egg get created. If it's not already familiar to you, the word gametes, that's how you say that, gametes, is basically just the unisex term when you don't want to specify sperm or egg. Gamete applies to both. After we learn to work some basic genetics problems, we'll dive into exactly what that production of gametes where segregation happens, exactly what that looks like. So just that idea, those two ideas, that's called Mendel's law of segregation. I know you kind of know this stuff from previously. I think where we take off from what you know, though, is starting to think in terms of the physical world. That these flowers that you didn't even think of as sexually reproducing individuals before, what they are doing, what these F1 parents are doing is producing sperm and eggs, about half of which get the big P and about half of which get the little p. What a Punnett square, which we're going to look at here in a minute, but understand what a Punnett square is. Punnett square is not prediction. It is not telling you what the kids are going to be. It is telling you what the odds are, probability-wise, as far as which sperm meets which egg, right? Because it's basically a crapshoot. It's basically probability. It's a roll of the dice. Maybe the big P finds the big P. That's going to happen about a quarter of the time. Big P finding little p, or little p finding big P, that's about half the time. Little p finding little p is our remaining quarter. Does that make sense? Sounds weird, but thinking in terms of sperm and eggs with Punnett squares is going to get you to the next step. So kind of try to keep that in your brain. Any questions? All right. Some quick vocab, if you don't already know it. Feel free to, I mean, if there's parts of this that you have down pat, you're never going to forget, ignore it. Don't waste time writing it down. But alleles, that's an important one. Alleles are the different versions of the same gene. So flower color in pea plants for us comes in two alleles, the white allele and the purple allele. Same gene, just different versions. When we are talking about what your alleles are, which two alleles you have, right? The two you got from your parents. Well, okay, are they big B, or big B, little b, or little b, little b? That is what is called your genotype. Your genotype is, hey, which two letters do you have? And we use those words you already know. We say if you've got two of the same letter, you're homozygous, but then we have to specify dominant or recessive, right? You're homozygous dominant, two big letters, homozygous recessive, two little letters. Then there's two different letters, big and little, that would be heterozygous. Which is great if you can figure it out. There are times when either you can't figure out the genotypes or you don't need to. You just want to find out the way the kids are going to look. Who knows the word for the actual visible version that you're showing? Phenotype. Phenotype, not with an F, but with a PH, is what you show. What do you look like? What color is your hair? How tall are you? Doesn't always have to be completely visible. Blood type, which we'll talk about before long. The phenotype, if you're type A or B or AB or O, those are phenotypes, even though they're not visible. You can make them visible with a blood test. And finally, we mentioned it and sort of defined it. Gamete is that unisex term for a sperm or an egg. All right, almost done. This is going to be review for a lot of you. Let's talk about how to set up a Punnett square. And we're going to write everything down. Again, skip stuff that you can skip if you want. Pay attention at the end. We'll refer back to sperm and eggs again, and it'll be important. So, first step, pick a letter. You got a genetics problem you're looking at, and a letter isn't given to you, pick a letter. It can be any letter. It does not matter what the letter is, except it's fairly common to pick the first letter of the dominant trait if you know which one is dominant. Not saying you have to do that. I'm saying it's probably a bad idea to do the opposite. You probably don't want to pick the first letter of the recessive trait because you'll get confused. Second, some letters are better than others as far as telling the difference between capital and lowercase. B is great. O, not so much. C, I love C. I like to pick ones when I can that look very different in upper and lower cases because my writing's not awesome and I get confused. Second, of course, you use capital for dominant, lowercase for recessive. You might not need to write that down, but let's say we're looking at big B for brown hair and little b for blonde hair. One thing I will say that I think can probably help you, and it's going to seem a little kindergarten-y, but I always write that down at the beginning of a genetics problem. I always write down which letter means what. It's a really good habit to get into for when you start having to use like Q, and Q isn't the letter that either one of those traits starts with. When you get into real weird traits, then you can't remember which one means what. It's really good to have that written down and just look back at it, so you don't have to stop working. Okay? So pick the letter, write down what's what. If we're looking at, say, two heterozygous parents who are brown hair, we make a Punnett square. The Punnett square is 2 by 2. And we put one parent's alleles above the square and one to the left. What do I mean by one above and one to the left? How do I set up this Punnett square? Walk me through it like I'm 4.
You put the letters at the top, like separated B and
Like that?
no, per box.
Per box. So you're saying you want one here and one here?
So do the same thing. Opposite side, the left side, but going down.
But vertically. So one here. So I want those separate? Why?
Because it's separate. Yeah,
Because when you make gametes, Mendel's law of segregation says the big B and the little b come apart, and one or the other, not both, go into the. These aren't the parents we're writing down here. What are they? In. That's not mom and that's not dad. Those are mom and dad's separated alleles, so these must be what? Sperm and eggs. That's what they are. These are the 4 different ways that a sperm and an egg from these 2 parents can come together. That's how I want you to be thinking of this. I know it's weird, but it will help. Each of these represents a possible gamete the parents can make. The outside of the Punnett square is possible sperm and eggs. The inside is how they can meet up. Inside is 4 possible genotypes of kids that could happen if those sperm and those eggs meet up. Does that make sense? Good, I can stop talking now. Nithya.
Parent get 2 recessive genes, they would just use the lowercase b.
2 lowercase b's, absolutely. And really, technically, you'd only need 1, right? Because they are the same. So you could cut your Punnett square in half if you wanted. Yep. Punnett squares, I know a lot of you are like, ooh, I loved Punnett squares. And that's cool. And we're going to do them. But we're not going to do them for very long. We're going to move beyond Punnett squares pretty quickly because they're a fairly simple tool that's only of limited use to us. But let's do some. Here's a set of genetics problems for you to play with. So, you're going to do 1 through 5, which is the whole front side and 1 on the back.